Yeah, I'd like to welcome everybody to the first Biophysical Society Cryo-EM subgroup webinar, which we're having during Biophysics Week, for which there are a lot of other events going on, both in the real world and here in the virtual world. And today we're extremely lucky to have with us Bridget Carragher, who's the technical, everybody I'm sure in the audience, Bridget will need no introduction, but Bridget is the technical director of the Zuckerberg Imaging Institute, which started up last year, and for which there's a lot of very exciting work going on in the Cryo-ET world. Just a very quick intro to Bridget's bio. So Bridget got her PhD from Chicago in 1987, worked in a variety of different positions in both industry and academia, and founded Nano Imaging Services, the company Nano Imaging Services, in 2007. And as I mentioned, she's now the technical director of the Zuckerberg Imaging Institute since January 2023. But really, on a personal level, Bridget has just made so many contributions to the Cryo-EM community that they are innumerable, so I'm not going to enumerate them here, but just massive contributions in terms of methods development, automation, both software and hardware contributions with Legion-on and Spotted-on and too many different things to list. And so we're extremely lucky to have her here to present us a seminar on the future of Cryo-EM and Cryo-ET. So just some housekeeping. So feel free to leave questions in the chat. We'll have hopefully 15 minutes for discussion after Bridget's talk, or just put your hand up at the end of the talk and any questions can be addressed then. And I'd also like to thank the committee of the BPS Cryo-EM subgroup for helping organize this, and in particular Pilar Cosio, whose idea it was to invite Bridget and who has been instrumental in making this happen, and I'm very glad that it did. Okay, so without further ado, Bridget, the floor is yours. We're really looking forward to hearing your talk.
Thanks very much, Ollie, and thanks, Pilar. I'm honored to have been asked to do this, and somebody commented earlier today, you must really be getting old when they're asking you for these retrospectives and perspectives. I think that's true. And indeed, I am getting old because I've been here in this field a long time, 40 years really. And I'm going to go through a little bit of the history because Pilar and Ollie suggested that would be fun for the new people to the field. And then I'm going to talk about some of the challenges that I think remain in single particle cryo-EM, and then also talk about the new stuff that we're trying to contribute to at the new Zuckerberg Imaging Institute. So I'm going to talk about cryo-EM, but as we all know, that's really two different areas at the moment, highly related but different. And the first is single particle analysis, which is where we get the exquisitely high resolution structure of these individual components of the cells. And that's been done largely by Cryo-EM at the moment, but a long history of X-ray and NMR. And of course, AlphaFold is now making a massive difference to that area. But then the other area is cryo-electron tomography, where we really want to know at high resolution the structure of these molecules inside the cell. And that's really the next frontier. So just a little bit of history. The beginning of this field for many of us was the 3D-EM Gordon Research Conference in 1985. I was there. I was lucky enough to be there as a student, and there were three other very important people there, which were the Nobel laureates who got the Nobel Prize in 2017, basically for the field of cryo-EM. And that was a fabulous meeting and continued to be a fabulous meeting and continues to this day, and made a very big difference to the way the technology developed for the field. So, that was the start of cryo-EM. Here we are back in 1985. And if we look at the growth of cryo-EM, one measure of that is the number of structures that were contributed to the protein data bank by field. Well, you can see in 1985, there was exactly 0. no structures were in the protein data bank. And then finally, in 1991, there came one. In 1996, good long time later, right, 10 years later, another one. And then there was a little bump up in 1999. A few structures started to be contributed, but it was pretty flat, not much happening until in 2013, the field took off with exponential growth and is now, chasing even X-ray crystallography, which has such amazing throughput and does such amazing work. But EM is now chasing that because it can do all these amazing things. So what happened in 2013 was what became to be called the resolution revolution, because it was so revolutionary. EM went from a backwater niche group to really dominating most of the new structure, contributing most of the new structures that came out. So that resolution revolution, our group was at Scripps Research Institute at the time, and we were very lucky to be knee deep in it because we got one of the very first new cameras that contributed to this revolution at the same time as Nico Grigoriev came to spend a sabbatical year with us. So he did some of this early, him and the group did some of this early work using these cameras. And the point of these cameras was that they took movies. They took better images because we didn't have they directly collected electrons, not having to convert the electrons to photons, but they also took movies. And so for the first time, we could see what is happening to our images while we take those images, while we expose this ice layer with proteins embedded in it to this high energy electrons. What happens to the proteins? And here's, the edge of the carbon hole across which the proteins are suspended. And we're looking at a few of these large rotaviruses. And I think you get a feeling they're moving. There's a lot of movement going along. And by the way, these are averages of 10 at a time. Otherwise, the images are so noisy, you can't see anything. So if we have all these different frames, then we can actually measure frame to frame to frame the movement of each of these each of these viruses. And these are exaggerated vectors, but this is a realistic scale. So many of them are moving by more than 25 angstroms. And so it was little wonder that the field had struggled and struggled to get to any kind of thing past blobology. We were famously called blobologists by the X-ray crystallographers, and quite right, because we were reconstructing blobs. And that was because our images were entirely blurred by that motion that was happening. And before the movies came along, before those new cameras, we just had one solid frame. All of those were just averaged together into this big blurry mess. And now that we had the movies, we could take out the movements in between and get these nice, crispy, clean images. And we showed very soon after that, in 2012 already, that you could do much better resolution structures when you did this cleanup of the images. And then very shortly after that, we started using it for real biology. This is an image of the HIV trimer before correcting for beam-induced movement and after. And you can see this was like night and day for us. I mean, there's nothing in this image of use, and here the little particles are, and as a result, we could start to do reconstructions of these incredibly important biologically relevant structures that were inaccessible to X-ray crystallography because they were too big, they were too floppy, or there were other complications with crystallography. So that was the resolution revolution, but it's important to note it was an evolution as well. Although the camera tends to get all the credit, a ton of work in that 30 years or so that transpired between 1985 and 2015 were a ton of work went into the microscopes. These microscopes we have these days are modern marvels. They are so stable, acoustically and electrically and vibrationally. These things can take angstrom resolution data for weeks on end without going wrong. And that's really quite an engineering feat. At the same time, we had a revolution in computers. When we first used these cameras back in 2012 or 13, we decided to try and get below 3 angstrom, and it literally took us months and months and months of computing on CPUs. Nowadays, of course, we have these GPUs, and we can do that months and months of computing in a few hours. So that has really revolutionized, and this came for free for us. The gaming industry, and now, of course, AI, has driven the revolution in computers, whereas this is all on us. Only electron microscopists use microscopes and these fancy cameras, and that's what keeps them expensive and keeps this very cheap. So in general, everything became better and faster, or better, faster and cheaper. So the whole revolution in cryo-EM relied on all of these. Without the good microscopes and without the fast computing, we would not have been able to make such good use of these cameras. And at the same time as the hardware was making these amazing advances, the software as well, data collection, processing, analysis, reconstruction, all got a lot smarter. And in that, when, by this time that we're collecting data, we're collecting 10,000 movies a day. When we first started doing this field, you've got 50 films a day. And even, later on, it was 1,000 images a day. Now it's 10,000 images a day. And then the software for sorting out these messy little particles and making sense of them is incredibly sophisticated, automated, and effective in terms of sorting out the particles that are in one conformation from another. So all of that got a lot better. So here we are in 2024, and all of this is now quite accessible. If you have a great protein that behaves well, you can get your high resolution map, and it's not going to, and all of this hardware and software works for you. But there is still one remaining problem, and that's specimen preparation. It remains a sort of a linchpin of single particle EM that trips people up some of the time when they're getting started. And I wanted to discuss two aspects of specimen preparation that affect us, one of which is the thickness of the ice that your protein sample is embedded in, and the other is the preservation of that sample within that thin film of ice. So ice thickness is important just for a very simple reason, that's data collection speed. The way we collect data these days, which isn't that recent, it's only 2018 we started to do this, but we lumber our mechanical stage across to a position, and then we do some focusing and stuff, and then we rapidly, with image shifts that are very, very fast, hammer out a whole set of images. We shift here, snap, 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 and we snap all these images up as fast as we can. We can get 50 targets for every lumbering movement of the stage, which takes time and effort. But if the ice is, and that gives us about 400 movies an hour, say, but if the ice is too thick or too thin in most of these holes, then we only get a few images for every big movement we have to make. And the ice is often too thick or too thin. And if it is too thick or too thin, don't bother shooting a picture of it, because you're going to throw it away later. So you're just wasting your time. So the difference between this, good ice everywhere, nice thin ice that you want, and bad ice is about double the speed of data collection. So this is much faster if you can control the ice, and because you're only going to use one Krios instead of two Krios, it's incredibly much cheaper, because these are very expensive instruments. So ice thickness is key, and it's hard to control. Here's an example from when Yongzi Tan, who's now in Singapore, was a grad student in our lab, and he was looking at a very challenging system for that time, a 45 kilodalton integral membrane protein. And he's making grids. These are grids he made on the same day, within 10 minutes of each other, same sample, same instrument, same everything. And this one looks very similar to that one. And then we zoom in. Yep, that looks about the same. And we zoom into the high resolution. And these are these messy little integral membrane proteins in the ice. And then this particular grid yielded a beautiful 3 angstrom map. And this one yielded an ugly blob. And then really the only difference between those is ice thickness. In this particular grid, the ice is exquisitely thin, about 20 nanometers, which is about the best we've ever been able to do over those size holes. And here the ice was all over the show and scattered around, and we just didn't get enough data to do the alignments and get a reconstruction. So it's hard to get that ice thin and controlled. And then when we were at NYSBC at Simon's Electron Microscopy Center, Eugene and Cassahoon did a systematic study where we got ice of all kinds of thickness and then systematically just divided it into thickness realms and showed how bad it would get if the ice became too thick, except if you're on a very, very good instrument. So ice thickness is key for data collection, speed, and quality. And I hope one day we'll be able to control that with customized grids. So Chris Russo and Katerina Najdenova, some time ago, 2020, announced this beautiful monolithic grid that they could make, where the grid bars and the substrate and the holes were all made in a clean room, and you could control everything about that. So I'm hoping one day that'll mean we can control ice thickness a bit better. especially if we start to play games on the surface of these grids. But a word of caution, at the time they said that, then Quantifoil licensed that technology and announced that they were going to provide those grids. And that was in March 2021. Whoops. And just yesterday I checked what's the current status of that. And the first pre-production batches, this is 3 years later, are now available. So this illustrates not that quantifoil is slow, but that it's very hard to do these things. To do them at scale, to do them right, to do them in large quantities is quite hard. So I think a lot more work is needed in this arena, and I encourage other people to go into it and think about it. And so that was ice thickness, but then sample preservation is also a key to data collection speed. If we asked the question when I was in New York, how many images do we need if the protein is very well behaved? And in fact, for 10 images, you can get 3 angstrom resolution. And these aren't 10 best images. These were the first 10 images to come off the microscope that day. So if you've got a great sample, 10 images is enough for 3 angstrom map. And 10 images only takes 2 minutes of data collection. So we would be a very high throughput technique indeed if this would be the case. But, a more realistic sample, more typical circumstances, and I'm just taking an example from a lovely paper that Rado and his group did some time ago, where they did a whole bunch of conditions, and they were looking at a GPCR. And they were doing it under different conditions, doesn't really matter, it's just illustrative of numbers. And they were getting 3 angstrom resolution. I beg your pardon. But they weren't using 10 micrographs, they're using 3,000 to 5,000 micrographs. And out of those micrographs, they were selecting millions of particles to do the averaging, but they were throwing a lot of them away. Only 5% to 20% of those particles actually contributed to the final map. So if we could contribute all of the particles to the final map, instead of 3,000 to 5,000 in micrographs, we could collect 300 to 500. And that's, these days, an hour or so of data collection. So again, we would get to higher throughput if we could control some of these things. So you may ask, and it also would be a lot cheaper because instead of 5 Krios time collecting, you've got 1 Krios collecting. So why? Why are we throwing away a lot of the particles, and what is going wrong? Well, we all know there's immense challenges sometimes with grid preparation when we're making cryopreserved grids. Sometimes the particles aggregate horribly. They look fine on the gel, they look fine in negative stain, they aggregate horribly when you put them into, when you vitrify them. Sometimes they adopt a highly preferred orientation. They're in one orientation only. You can't see the other views. That's hard to do a 3D map from. They might not go into the holes. They'll stick to the carbon. They won't go anywhere near over the open ice where you want to be able to see them. They may be partially damaged, or they may be completely denatured. You can see your protein in stain. You put it into ice, you can't see anything. And the reason for this is something that Jacques Dubochet, in the very, very original paper in 1982, when he discussed vitrifying grids, it had one sentence that said, the fact that in the thin film method, a thin layer of ice that you suspend your particles in, the particles are always close to the surface, has significant consequences that have hardly been investigated. And that was absolutely true, and we didn't investigate them for 30 years after that, I think, until we really started to, until the resolution revolution came along, we really started to care. And Bob Glaser recognized this many years ago, and it was, telling all of us that the air-water interface is going to cause problems. And the particles don't want to be at an air-water interface. And then Alex Noble in our group in New York did a whole bunch of tomography on single particle data. So he basically had a single particle grid, and then we tilted it back and forth and did a tomogram, did a 3D reconstruction. And he did this for many, many, many different samples that happened to be in the lab at the time. And if we just zoom in on a few of those 50 examples he did, what you see is that indeed, for almost every single sample we looked at, the particles are stuck at either both air-water interfaces or one of the air-water interfaces. And there's a few particles, that are actually in solution, but the rest are glued up against the air-water interface. And one can imagine that very hydrophobic area of the air-water interface is not going to be a happy place for proteins, and all kinds of things may go wrong with them. They may denature, they may adopt a preferred orientation, they probably aggregate and do all kinds of horrible things. So people have considered all kinds of ways for trying to get around the air-water interface effects. And one of the projects we did in New York was called the Spot-it-on project. And in this project, instead of applying a large volume of liquid to a grid and then blotting it away with blotting paper, what we did was use a piezo dispensing device to spit very tiny volumes of sample, picoliter droplets of sample, at a grid as it flew by the spraying head. And as a result, the droplets were sprayed onto the grid in a line as the grid went past on its way to the liquid ethane. We got the sample to spread out without the blotting paper by having what we call self-wicking grids. And this is really just a grid where we grow a huge lawn of wires, copper wires, on the copper surface, and then we have the substrate underneath. And what happens is that the water arrives on the substrate, starts to spread out, but the instant it comes in contact with those wires, they act like a capillary action, just pulls the water away into a thin film. So why does that work to ameliorate the effects of the air-water interface? Because the particles don't spend so much time in the thin film. So the drop arrives, spreads out, and 100 milliseconds later, it's everything stops. Whereas on a Vitrobot, it's seconds of time between you blotting this, and then you take the blotting paper away, and then you drop the grid. There's seconds of time that the particles spend in the air-water interface, and as a result, it does help. So it helps, prevent preferred orientation. It helps prevent denaturation. In this particular example, this FACT, which is a machine that unwinds and rewinds nucleosomes, we could never see anything in ice if you didn't use Spot-it-on or Chameleon in this case. You had to use this, otherwise it was completely denatured. So it helps a bit. And we know we want to keep our particles away from the air-water interface. So Spot-it-on or Chameleon, which is the commercial product for it, might help. People try detergents, people use graphene substrates, other substrates, they use sacrificial layers, enclosed systems have been proposed. There's a lot of different things we could try, and none of them right now is a golden bullet. None of them gives you the answer for every single protein that walks in the door. So again, this is an untapped area, and one I think that needs to be worked on if possible. Because if we do control that problem, we will get better data. Instead of the 5% of particles, maybe there's 100%. That means we can get data faster, and that makes Cryo-EM much cheaper and more accessible and more high throughput. So that's the better, faster, cheaper. What about smarter? So where smarter came in for us in New York, at least, was that the single particle pipeline has a bottleneck for grid screening. Once you have a perfect grid, you're probably 24 hours away from a high resolution structure. But to get that perfect grid, as you saw, things aggregate and preferred orientation, all kinds of horrible things happen. And you can fiddle with that by trying different things with your sample. You can change all kinds of things with your sample. You can change all kinds of things about the grid that you're suspending your sample on. You can change all kinds of things about the blotting device. And if you're using Spot-it-on or Chameleon, the plunging time and the grid batch, etc. And sometimes it feels like maybe the phases of the moon also have quite an effect on how you're preparing your sample, because it's not very predictable. So that's a bottleneck, because you have to go and look at those grids. You make a grid and then you have to go and see, well, did that help? And how you do that to assess that is you have to go put that grid in a microscope and take a whole series of images, because you want to assess the ice thickness across the grid, the distribution, the number of particles that you see, what the particles are, how they distributed across the whole, whether they have preferred orientation. All of this needs to be assessed. So you have to put a grid in and get all those images. Then you have to go to the next grid and get the more images, and another grid and another grid. And people can go through dozens of different trials and error to try and figure this out. So that we felt needed to be fixed and automated and taken out of the hands of an operator. Well, you might ask, well, isn't data collection already automated? We've been automating this for 20, 30 years now. And yes, it is, but in general, the algorithms that find the squares, so if you look, low resolution, you got to find the squares, then you go up in low magnification, up in magnification, got to find all the holes. So there's certainly algorithms that can find squares and holes, but they tend to be heuristic algorithms that require tuning parameters. So once you can find, you tune a few parameters here, you can find the squares, it'll find the squares on the rest of the grid. But the next grid over may look quite different, and a new grid may look different again. And similarly for these holes, an algorithm that can find these holes very well may not find these holes very well, or these, because they're too faint and it's very hard to see. But people are very good at this. People can say, yes, I see those very well. I can find them easily. I know where they are. So if people are good at something and will annotate it, machine learning is the obvious answer to how to develop an algorithm to do better. And so in the data collection software that we developed called Leginon, every image we ever collected is in a database. And it's linked to its higher magnification images. So this low mag image is linked to the next one up, is linked to the next one. So basically we have an annotated set of images where we know where the good data came from, because we can see this is good data, so that was a good hole, and this is a good square. So that's the basics of training a machine learning algorithm. And Paul and Kristen, who were in the Simon's machine learning group at SEMC in New York, developed a system called Ptolemy. It's all been published well now, where they had a machine learning, a CNN classifier for the squares and a UNet for the holes, and a bunch of other traditional image processing, so that you could find all the squares, of course, and then decide who are the good squares. That's a bad square, that's a good square, etc., etc. And the same with the whole localization, and also you can measure thickness to evaluate whether something's a good square or a bad square. So this system, Ptolemy, in collaboration with Leginon, the data collection software, means now we can screen grids automatically. So the grid screening says, okay, you have a grid, find me 4 diverse squares. I want different kinds of squares because I want to know what's on that grid. For each of those squares, find 5 holes. And for each of those 5 holes, take 5 high mag images. And then on to the next one. Auto load the next grid and go and on and on. And so if you test that auto screening, this is the auto screening, the machine learning algorithm, against what a human operator would take to screen, say, 11 grids. Humans take between 5 and 7 hours to do that at the microscope for the most part, with few pauses in between. And the human time required to do that once you have the machine learning algorithm is 10 minutes. So it's a huge saving in operator time. It can be done, of course, over the night, over the weekend, etc. So that's the time is important. The machine learning algorithm also, in at least the trials we did mostly better than the humans in terms of how many good targets it picked and whether they were suitable. So instead of having to sit and set up an algorithm each painful time, you can just sit and watch the data stream in and decide which are the good ones. And I think that's used routinely now in New York to screen grids. And so what we really want, of course, is to close that loop. So we can screen the grids automatically, but what we really want to do then is go process those grids and then extract metadata from them and learn from that. Where is the even better real estate that we should be going after? And that's sort of underway. It's quite a hard task because it's hard to know exactly how these measures relate back to the data collection parameters. But that is something I think also is will come up more and more in the future until data collection becomes an entirely automated process. Okay, so that was single particle analysis. We know a ton of stuff about the individual components of cells, but that's like, knowing the components of a watch and expecting to know how the watch operates. In order to really know what is going on inside a cell, we have to look at the machines in situ, in their native environment inside the cell. And the only game in town at the moment to do that at high resolution is cryo-electron tomography. And so that's the focus of many people's efforts all over the world. And for us here at the newly created Imaging Institute, Chan Zuckerberg Imaging Institute, that is our mission, to try and understand the architecture of the cell at high resolution. And the first grand challenge for this Imaging Institute, there's planned to be 3, is to understand the molecular architecture of the cell at near atomic resolution using cryo-electron tomography. So we want to develop tools and technologies to make the cryo-electron tomography pipeline easier. So that pipeline is very complex. It's a whole series of complicated steps. You need your cells. You probably need to label those cells. You need to say, you need to identify the protein that you want to look at. You may need correlative light electron microscopy, probably super resolution, in order to be able to find where components are interacting and to be able to slice a thin section of that cell open, which is what you're going to need to do cryo-electron tomography. So you need a focused ion beam scanning electron microscope to slice out a thin slice of the cell, hopefully at an area of interest that you care about. And then you go off and you do the cryo-electron tomography, you take your tomograms and your tilts, then you have to process them and account for various artifacts. Then you have to pick out individual particles from these noisy 3D slabs and average those together to get a high resolution structure, just as you have to do in single particle microscopy. And then interpret, integrate, model. So all of this requires technology that's better, faster, easier, and smarter. All of this needs work right across the pipeline. And so our defined goals for this first grand challenge are to develop robust, reproducible, openly accessible tools, very important, and to improve some of these aspects of cryo-electron tomography, and then apply that to key biological problems, because we need to keep ourselves honest. So just to explain the goals we've set ourselves the goal of improving contrast, resolution, and throughput. So what do we mean by contrast? Right now, in these very noisy tomograms, we can mostly see ribosomes, these nice big black blobs here, noisy fuzzy blobs. Those are ribosomes. We can see those. Everything else looks like a tiny little noisy dot, other than the membranes. So really, if you say the percentage of the observable proteome, if you divide it by mass, if we're up here by ribosomes, we're seeing a fraction of the observable proteome. And in fact, we're seeing less than that, I think, because if you look at the number of high resolution structures that are actually contributed by this, it's even less than that, less than it should be. But that's because the method's hard. So what we want to do is push this curve. We need to push ourselves up this curve using a bunch of tools and technologies that we think will help until we get to this point where we can start to see 100 kilodalton membrane receptors, in which case we'll be seeing basically 90% of the proteome. So we have to improve contrast so we can see what's in these noisy images. It's also quite a heroic business doing a 3D map from subtomo averaging at the moment. This is the beautiful work from Yulia and Dimitri, where I think there were 20,000 ribosomes were averaged from subtomo, from subtomograms. And that's sort of doable now, 20,000 copies of them. So that's kind of doable. But if we go to smaller and smaller structures, it becomes harder and harder and harder because we just need more and more and more copies of these proteins, and it becomes infeasible pretty quickly. So the goal in increasing resolution is to drag that curve down so that even for membrane receptors, we get into this doable range. And we believe there's a bunch of technologies that can do that. And then finally, throughput. These days you see beautiful data being published, beautiful structures being published from data that was acquired 4 or 5 years ago. So, the throughput of the system is really terrible. If you look at a timeline, we can get data collection maybe, a few days or so and processing. But to do the annotation, the full subtomogram averaging can take on the order of a year. So that's really a demonstration and not an experiment. An experiment needs to be done something like the order of a month. You want a hypothesis, you want to go do the experiment, you want a result, and then you want to say, okay, that means I'll ask this next question. So we really need to compress this whole timeline. Whoops, sorry, I've lost my cursor. Compress this whole timeline into a much more feasible length of time. So those are the goals we've set ourselves. Obviously, any improvement in contrast and throughput will automatically improve resolution. And to pursue those goals, we've defined a whole bunch of technology research and development projects. And these are them, but I'm only going to talk about a couple today, highlight a couple that we're actively working on. And the first of those is this incredible device that Holger Mueller's team has built at UC Berkeley called the laser faceplate. And the laser faceplate is indeed a faceplate. I think many of us experienced the Volta faceplate some years ago, but it didn't quite pan out the way we wanted. And the laser faceplate is a very intensely focused laser beam that the electrons pass through that does the phase shift. And this is the proof of the pudding that they published a couple of years ago with the laser off and the laser on, and you can see this enormous leap in contrast because of that phase shift. It's even more impressive if you look at a very thick slurry of sample. Here's Rubisco in a giant, big, sludgy mess, and with the laser off, you can see nothing. With the laser on, I think I can see the shape of those Rubiscos in there. And this is more what we're facing with tomography, where we're looking through a big, crowded wodge of proteins. So this is sort of very exciting and very hopeful. So that's what Holger's done. And this is what his lab looks like in Berkeley. It's incredibly impressive and awesome looking site, but it's not for the faint-hearted, and only a laser optics physicist can be very happy there. So our job at CZII is defined as taking this giant thing and shoveling it inside a standard electron microscope and making it very easy to use. So that's been a very complex project. It's a complicated collaboration between Thermo Fisher and UC Berkeley, us and Chan Zuckerberg Initiative itself, and then an engineering company who we've hired to do the design called Applied Precision Design. So that's our goal. Here we are. This is the progress we've made with it so far. Thermo, and with Holger's help and everybody contributing, designed a new column, column for the Krios that would have room for the laser faceplate and, incredibly enough, improved the quality of the imaging parameters, reduce the chromatic aberration. And the most amazing thing about this is it's a better column with more room, and it came in a year earlier than the one we would have had without this. So that never happens. Usually, if you improve something, you wait another year. But in fact, this has moved it up by a year. And then Applied Precision Design has designed all of the optics board and the laser faceplate and the arm that it'll have to go on. And all of this has been miniaturized and crammed in so that it will fit inside the Krios column and not get in anybody's way. That wasn't easy to do. And it's very hard because of the dual laser faceplate. There are going to be two crossed lasers. And so there's two of everything. You saw Holger's enormous lab that's for a single laser faceplate. Now this is a dual laser faceplate. So there is a heck of a lot of miniaturization going on there. And this is the actual dual laser faceplate. The 4 mirrors inside here, exquisitely perfect mirrors. If they're not perfect, then any speck of dust will cause them to heat up, and then you'll lose the resonance in the cavity, and the laser faceplate won't work anymore. So it's a complex system. We think it's on track. The instrument itself will be delivered at the end of June. We hope to get it all working sometime by the fourth quarter of this year, and hope to be able to present some results to the community. Wish us luck. It's a very complicated project. Okay, so that's one project. Another project that we're working on is just the entire processing pipeline, like of course everybody is. And the goal there is, this is what we collect at the electron microscope. These are the tilted images. They're wiggling, they're jiggling, they're moving around. The whole thing is like a big sloppy bag of jelly that's jiggling as you collect the data. And that has to be turned into a nice block, rigid block of well-aligned, corrected 3D data, out of which we have to find all of these individual particles and then average them together to do subtomo averaging. We'd love to be able to do that for many particles. Right now, it's being done for the ribosome, and at least at high resolution, and not much else. So there's a huge complicated pipeline that does all this, and almost every step needs to be improved. So how we think about this, I'm nearly done here. I think I'll finish up pretty soon. How we think about this is 4 steps of the pipeline. First is the pre-processing, turn those raw frames into a 3D volume. Then we've got to find the particles. Then we've got to do subtomo reconstruction and atomic annotation. All of those have to have metrics measured so that we keep ourselves honest and start improving. And it needs to be wrapped in a workflow so that it's very easy to use. So let's just talk about the first step, the pre-processing. That's a whole bunch of different steps: motion correction and magnification correction and correcting CTF and a bunch of different things, and denoising. And our goal there is to do this in real time, by which we mean as fast as we can collect the data. So we've made some progress there. Sean has sped up the RTOMO workflow. Ariana has done a beautiful job on denoising and made that 20 times faster, so that as a result, we can do this, from raw data into a 3D volume at a speed actually faster than we can collect the data. The denoising is going very well. Ariana has a beautiful noise-to-noise algorithm working. It's very fast. And this is the sort of night and day difference we get from that. The contrast improvement is enormous when you do this denoising. We can see these individual proteins. And it works also for cellular material rather than lysate. Here I think, yeah, that looks like a nucleosome, which is here, but it's much harder to have identified it there. Okay, so the other aspect of the pipeline that we really need to address is the annotation. We can get this done now in days or real time. This can probably be done in days, especially if we have access to enough compute power, but this takes many months at the moment. Annotation of the images is by far the major bottleneck, and that's why our view of the cell, instead of this nice rich environment, is the membranes and the ribosomes, essentially. So annotation is hard. It's a very crowded environment. It's very noisy. The contrast is low. We hope to improve all that. And we probably need tens of thousands of these particles to get to atomic resolution. So there's various methods. Unfortunately, there's template matching, there's machine learning. Unfortunately, a lot of the time it's done manually. People sit there and pick out particles because these methods aren't working well yet. So that means annotation can take weeks to months to years. So to address this, what we think is we need much better machine learning algorithms, because if people will hand annotate, then the machine learning algorithms can learn from that and improve it. So to address this, what we've done is put together a database of annotated tomograms. Right now, there's 13,000 tomograms in that database. If you can read the little QR code there, maybe it'll come back later when the movie's over. Then you can go and browse through this data. You can download this data, you can download the annotations, you can do anything you like with it. We're working closely with EBI on that project, and we hope to keep adding to this database. What can you do with such a database? Well, what Utz recently did with it, he took the 13,000 tomograms and he ran the membrane algorithm from Ben Engel's group and annotated all 13,000 tomograms in a matter of a few days, with quite a bit of GPU power, but still not infinite GPU power. So originally we only had about 1,000 of the tomograms annotated. Now all 13,000 are annotated, at least with the membranes, and we hope to add particle picks to that shortly too. To support all this, we're going to organize a machine learning challenge, which we hope to announce sometime in the next few weeks. So the Imaging Institute will provide the data and do the evaluations, etc. Kaggle is a major sort of machine learning competition organizer, and they will host the competition. We hope we'll get this done in the next few weeks, but it is hard. One of the hardest problems for us is, what is ground truth? Nothing except the ribosome is very, very well determined that we know goes to high resolution, and nobody wants to do the ribosome. They all want something more challenging. So it's difficult to come up with the ground truth, and we don't know how to evaluate the results in the absence of ground truth. What we've done is build a structured data set, so some cell lysate with known particles added. So we kind of have a feeling, or we have some knowledge of what's in there, and we're going to annotate that, and that we hope will be the basis of this machine learning challenge. Oh, just two quick little things then. We do have biological driver projects. We feel we must keep ourselves honest. We can't do this in a vacuum. We need to be doing biology in-house as well as collaboratively. And we have two projects going, one of which is to isolate organelles and then study those organelles, which is a much easier project than trying to study them inside cells, because we can get massive throughput. We get thousands of these tomograms on a single grid, all covered with lysosomes or mitochondria or endosomes or whatever we're looking at. If you look at these isolated lysosomes, and after denoising, man, you feel like you can start to see some of these proteins in there, the ATPases, and then who knows? Here's what people imagine mTOR to look like to imagine something like that is there. So there's a huge wealth of data that we can start to mine as soon as we have some of this annotation working. And then the other project that biological project that's in-house is to look at the primary cilia and the ciliary synapse that Xu Shen discovered, that cilia can synapse with neurons in the brain to alter transcription. And again, we're starting to see some very interesting structures in these cilia after denoising. So that's it. This is the group. We were 5 people a year ago, and now we're 30 or 31. We are hiring. There's still jobs out there, so we would welcome people to apply for those jobs and join us in this very grand challenge that we've taken on. Thanks for your attention. Oops. Hello? Anybody out there?
Thanks very much for that, Bridget. That was fantastic.
Hi, everyone.
And fascinating to hear about all the latest from CZII in particular. Does anybody have any questions to start off the discussion? Feel free to put them in the chat or just raise your hand. If nobody does for now, then I'll certainly start off. So one thing I'm curious about is, are you guys exploring at all the high resolution template matching approach? Like, are you collecting trying to collect high dose zero tilt images as well, like particularly for things like the lysosomes? That seems like that would be an interesting approach to explore.
we already did. Reza, who's been heading up all of that up, Bronwyn came to visit last week, and we got the exact conditions she wanted us to collect this under, and so now we have a massive data set that we will hand to her, or hopefully people can work on. We also hope to put all that data onto the portal at some stage so that people can play around and try these various methods out. So indeed, we do feel both approaches will be useful.
Great.
Gosh, there's 120 people here. There must be another question.
There's a question in. There's 2 questions in the chat. I don't know whether you want to. Do you want me to relay them, Bridget, or?
Oh no, I can read them.
You got it.
Yeah. So I think everybody can see chat, right?
Yeah.
So the question is Cryo-EM, and what do we think is the major challenges, and what are the plans for CZII? So I mean, the major challenge is that light microscopy and electron microscopy are incompatible resolutions, right? A pixel in a light microscope is your whole tomogram. So if you're trying to see if 2 things that are labeled are touching or something like that, that's very hard to do. We do think that super resolution light microscopy may help provide better localization. And the other, of course, major challenge is we have to cut a slice at an angle through a cell that's 200 nanometers thick, and the resolution of the light microscope isn't 500 nanometers. So, how are you supposed to hit that target? But 2 things we're doing. Delmic is bringing their new system here. I think it's being installed next week onto our Hydra. And they have a new cylindrical lens, and maybe that'll help. And then Zuckerberg Initiative has funded GoCore at Berkeley to build a super resolution light microscope to go into the Hydra. That's a longer term project. That's probably a year or 2 before that gets built. But so right now, you said, what is the Imaging Institute doing about it? Got the Delmic system, going to collaborate with GoCore, going to collaborate with Pete Dahlberg. So right now we're outsourcing that apart from the commercial Delmic system. But we're open to ideas as they come up. Shall I read the next one? Oops, sorry. Now it keeps. Ollie, can you do it, because it keeps bouncing?
Yeah, sure. Yeah.
To the
So there's a question. There's a question from Omid Haji Gassemi. I'm curious if the machine learning algorithm will become too biased in SPA towards thin ice, where in some cases thicker ice may be needed, particularly for large complexes. Is this something that you're incorporating into the feedback loop, I assume, in Ptolemy?
I don't know what the Ptolemy group is doing. I mean, I know for us, we're going to train on the tomograms, the thick tomograms. So the reason we didn't just use a mixture of proteins, is because they do go into a very thin film. But if you put them into a lysate with the lysosomes, then they're in a nice thick film. So we have things we know, but at least they're in a thick film. So it's emulating more exactly what a tomogram does. But I do think you're right. If you did only work on thin ice, you would have something that was quite biased, and you'd have to retrain on something else.
Okay, we have a question from Manoj Saxena. Wondering if you're planning to do tomograms for just the nucleus or for individual chromosomes. I assume targeted towards sort of genome architecture and that kind of thing.
Right. Not right now, but I suppose we could. We're very open to collaborations of all kinds. I mean, we're a small group, right? We look large, but we're only 40 people in the end, and we really want to collaborate with as many people as possible. So what we're going to build is pipelines to hopefully do this fast, at scale, rapidly. So if you come in with a sample, we can give you 1,000 tomograms and maybe annotated tomograms at some point, and you can go and do that biology. So although the nucleus was not in our list so far, I think it's a very good target, and we'd be excited to do that if somebody has a project that is ready to go.
Okay. We have a question from something. That one, the pronunciation is not on me. There are literally no vowels in that username. In the start, you mentioned that GPUs helped, but will workstations be enough for future Cryo-ET? What limits your pipeline computationally? Is it VRAM? What I guess addressing, what are the limitations computationally right now for Cryo-ET pipelines?
Right. I mean, they are large. When Utz ran this whole membrane right across 13,000 tomograms, he had 32 GPUs at his disposal. And that's because Zuckerberg initiative has put a big effort into making large GPU engines available to somebody other than, the major compute companies. They announced this a few months ago. They said, they think machine learning is going to be everything. You're going to need these large language models. You need the academic community and the non-commercial community to have access. So they're building a massive cluster. It's not built yet, but we have access to some early, a version of that, and so that's why Utz can get 32 GPUs to just race through 13,000 tomograms, and can race through 1,000 in a few hours. Do all of the denoising and membrane extraction, etc. Once we get to template matching, I think that slows down, and the machine learning methods are really slow. So that's when we think we need 100 GPUs all the time, running day and night, just to do the processing, and then probably another 100 to do the fancy, large language model training and things like that Dari Kamanias is doing for the refinement pipeline.
Great. So Chang Sun is asking, and feel free, anybody who's asking a question, feel free to jump in and unmute yourself and ask the question yourself if you would like. But Chang Sun is asking, what kind of resolution do you envision that we can achieve for 100 kilodalton membrane protein with Cryo-ET in 5 years? This is a crystal ball question, and I imagine it very much depends on abundance as well.
It depends on abundance. So it's a numbers game, right? We have to be able to average enough of these together. So it depends. Are you talking about in a native cell where there's 5 of them, or are we doing some purification like we're doing for the organelle project? But if you have enough of them, why not 3 angstroms? I'm an optimist. I wouldn't have been in this field as long as I have if I wasn't an optimist. So I do feel, we were blobologists, I don't know, 10 years ago in single particle, and now we solve things at 1.2 angstrom resolution. So I think Cryo-ET is ready for just such a revolution. I think it's going to take a lot of different things, better contrast with laser phase plates and denoising, etc. We need labels so we can find things easier. We need a million, well, not a million, but dozens of new technologies and advances. But then I do not see why that should not be possible in 5 years. I certainly hope it won't take 30 like it did for single particle.
Well, so just following up on that a little bit, so narrowing it down a little bit, you are limited in a certain sense, like the thickness of the lamella is going to be a problem, right, for things that are that small. And I guess that's where the improved contrast from the laser phase plate comes into play. Do you have any back of the envelope idea of, for something of reasonable abundance, how small can you go, do you think, with the kind of contrast that you can achieve with the laser phase plate? Like, how much does it help you?
So Chris Russo does have an idea. We've been collaborating with him, and he consulted for us to do a whole bunch of calculations as to where we might get. And those curves that we're drawing are based on his kinds of calculations. So I think we were cheating a bit on those curves because we mean complexes. So will we do 100 kilodaltons? Maybe, but maybe we're doing it in complex with two other things. Almost everything in the cell is in complex with something else. So maybe we get bulked up naturally because it's talking to one of its mates. So we maybe don't have to go down that small to still be able to see that 100 kilodalton protein, as long as it's got three things, surrounding it. So yeah, those calculations have been done, but they're theoretical. I'd like to follow it up with some experimental and see where the two meet before we put money on it.
Sure. Okay. So Sean Hansen asks, how do you assess the future perspectives of in-cell electron-dense fluorescent labeling of protein targets, something akin to gem tags? Thanks for a great talk.
Thanks. Yes, very much so. We have our own project for doing this. It's one of our people, Bisco Hill, is trying to work on a fluorescent gold label or fluorescent other structure label, so we can do both at the same time. But we think right now we're focused on just labeling the outside of the organelles because we want to find the mTOR or whatever on the organelles. But in the meantime, there's Chan Zuckerberg initiative funded, I think 10 or 12 or something, external grants for the community to work on a similar thing. So there were 12 or so proposals saying we think we can do this and we think we can do that, and those are being funded, and for a year. They were very short terms, like what can we do in a year, what can we see will work. So I think there's a lot of work to be done there. I think it's very hard, very difficult, to get the label into the cell, to make it specific, to get rid of the background, to make it non-toxic. All of those things are very difficult to do, but there's a bunch of people who are doing beautiful work on that, and some of them have been funded, and we'll see what comes from that.
Sort of relating to that, are you thinking about generic tags for subcellular compartments, like things that could be used for anybody who's working on mitochondria or anybody who's working on lysosomes, for example, to sort of make that more accessible? Because right now a lot of these tags are very bespoke, right? Everybody's doing them for their individual target.
Right. I think yes is the answer there, but I'm not the expert in this, so you should probably check with somebody who's a bit more knowledgeable. But as my understanding is, yes, the tag, once it's developed for the mito, will be available for mito. Developed for lysosome will be available for lysosomes.
Great. Zunlong K is asking, challenging but very exciting projects in Cryo-ET. Thank you for putting the grand team together for the grand mission. How do you envision individual labs fit into this grand mission? I guess this is talking about like collaborations with outside labs.
Yeah, we're very, very keen on collaborations and all kinds of outreach because we are very tiny, and we want to contribute where we can contribute. We don't expect to solve every problem. We're just going to do the problems we think we can make, like the laser phase plate. Very expensive, very difficult project. We can make a contribution there. But we will definitely have a lot of collaborations. We're going to have a fellows program where we hope to have, a postdoc or grad student, fund a postdoc and a grad student who embeds with us for a year or so. And then we fund part of the PI to sort of get, nurture those collaborations along. We have a visitors program. So we're just getting going. We only got into our building in November. So we're just getting going on some of those things and starting to think about them properly now. But those will all be announced sometime this year. And then we really, really encourage people, to come visit, to hang out with us, to, embed a postdoc or a student with us and, use the toys and use the pipelines and see what they can get done.
Great. So we've got one last question, time for one last question, I think, from Rubaya Rashid, which is, I think, targeted more towards like volume and tissue level stuff. So I was saying that Cryo-EM and Cryo-ET are useful tools to look at the 3D structure of single molecules, proteins, the cell architecture. But I'm curious if it's possible to look at a larger size range, to look at engineered biomaterials or tissues on the range of nanometer to micrometer scale. So I guess looking more at, can you put this together into a montage of a tissue or some more expanded region?
Right. And in fact, we're Grand Challenge 1 for the Image Institute. Grand Challenge 2 will maybe be dynamic imaging, and 3 was thought about deep tissue. So there is sort of a longer term view over the 10 or 15-year life cycle of this Imaging Institute. We personally are very interested in block-based imaging in cryo. We do think, it's untapped right now. There may be better ways to get contrast off that block face and really get some information out of it. We have a visitor coming for the summer who's going to work on some aspects of that. And then we eventually will also put a team onto it. Right now we're focused on other things, but block face was one of our technology research and development projects that is in our sort of roadmap that we would like to work on at some stage, because we're very aware that, in a tomogram, you're looking at a tiny, tiny portion of a cell. You need to scale up, and do that block face or, maybe deep tissue or whatever at some point. So, as you go down in those resolution ranges.
Wonderful.
And I see Steve Jett said 9 grants for the Clem label. Thanks, Steve. I was just guessing.
There we go. All right. So I think we've just about used up our allotted time, but that was fantastic. And everybody, please thank Bridget again. And thank you so much, Bridget, for joining us. This was wonderful and extremely informative. I enjoyed it enormously.
Thanks, guys. Thanks for inviting me, and thanks for taking the time to listen.
Yep. And everybody join the Cryo-EM subgroup for biophysics next year. We want to see all of you there. See you.
Bye,
Thank you. Bye.
Bye.
Bye. See you soon.